So thank you for inviting me to speak, and thank you particularly to all of the families who have come and traveled either close or far to be here. It's really, really inspiring to me, this community, and how our patient families are such equal partners with us in this endeavor. It really inspires me, and I know you've all been on a crazy journey through this diagnosis. You all started on a journey where you had concerns about your children, about what was going on, and people didn't have answers for you. And then the answers came, and glycobiology is really confusing. And this may not have been you, but this was definitely your physicians when they got that answer. And you have all moved to becoming glycobiologist experts with us, and certainly experts in your own children. And so I hope that having another research talk after many of you have suffered through research talks doesn't leave you feeling like this. But what I hope for those of you who have suffered through all the research talks, and for those of you who are here for today's session and hoping to learn some about the research that's going on, that I can provide a little bit of demystification around this whole topic of a CDG model, and that I can give you some highlights to take home and some clarification on where things are currently. So what is a model? So if you look it up in the dictionary, you'll see it's a representation of a person or a thing, and it's typically on a smaller scale than the original. And so here, a model we may all be familiar with would be a model of a car. But what do we mean when we mean CDG models? Well, we mean a representation of a person or a thing, a representation of your child's disease that we can then understand and use. And so we use CDG models to confirm new disease genes. We've heard a lot of discussion about that from HUD. We use these models to determine what these new disease related proteins do, and we use them to investigate what goes wrong when that protein isn't working, and to test potential therapies to try and decide, is this something that's going to work for your child? So there are different models to answer different questions. So if we go back to our example of this model car, this model car would be a good model for looking at how the different aspects of the car are put together. It may help guide us on relational size between parts of the model, but a stationary car model is not going to be a very good model if we want to learn how cars move. You might need to change your model. And this, as a remote control model car, may give you some more insight into that. But if you want to understand how a real car motor works in making it move, having the battery of that remote control car isn't going to tell you very much about a real car's motor. And if you want to understand what happens when a car is in an accident so that you can understand how to help in that, you need a very different car model. And so you may need something that's more the right size, but it may not actually need the engine that's in a normal car. So we have to understand what aspects we're trying to model so that we can get the right answers from our models. So when we come to CDG models and sugar, this isn't going to tell us very much about your child's disease either. So in trying to think about the different models we've heard about, we can sort of divide them up in a couple of categorizations. One is the material they're made of, and another one is how they're produced. So various materials, we've heard a lot about cell models, and then we've heard about model organisms that we may be able to use for understanding CDG, and even computational models. And then how they're produced, they can be naturally occurring, they may be genetically modified, they may be chemically induced. And so we'll go through some of these categories to try and understand what they are and what we've learned about them. So in the concept of naturally occurring models, it turns out that animals have genes, they have genetic diseases, and so there's actually naturally occurring organisms that also have CDG. So this is a picture of a CDG cow, in fact, that was known to be naturally occurring. And a couple of years ago at our hospital, we identified a human patient who had very similar features to this CDG cow, a naturally occurring animal that has the same genetic defects and much of the same changes in the glycobiology that would allow us to understand. But studying a cow is a really big animal and really challenging and really expensive. So there are a lot of other models we go to, and it really depends on what we're trying to model and how we're trying to answer those questions and which answers we're trying to get. So some of the most basic models go back to cells, which is really one of the most basic functional components of an organism. So our bodies are made up of cells. Those cells are grouped together into organs, and those organs are all put together into systems that form our bodies. Some of the nice things about cells is that they can really be grown in the laboratory. So as far as good things about cells as a model is that they're pretty easy and cheap to maintain. You can share them across labs. You can actually manipulate them genetically relatively easily, and you can then determine what that protein is doing in a cell. And we can even determine what is going wrong in the cell when that protein isn't working. But it's not a whole organism. It doesn't necessarily tell you what's going wrong in the whole organ system, what's happening in the rest of the body. And many of the cells models are actually derived from cancer cells. So we may also be looking at aspects of cancer when we're trying to look at these cells, and not what's going on in a typical CDG patient. And it doesn't tell you about other cell types in the body. During our scientific presentations, we've had a lot of cell models that have been presented trying to understand what's going on. So some of these go back to Dr. Randy Sheckman, who was our keynote address and Nobel Prize winner, who talked about how cells move their contents between the compartments. And this was especially relevant for understanding how these glycoproteins are moved through our cells. And that's something that's really the basic cellular biology that's relevant to all the CDG types. And then we had Dr. Taro Kinoshita, who talked about models for GPI disorders, and he used cells to confirm patient mutations as disease-causing and to test new drugs for these GPI disorders. And then we had Vladimir Lupushin, who talked about the COG protein biology, which is, again, how we move glycoproteins through our cells, and gave some really interesting insights into the fact that these COG CDG types have glycosylation defects, but they also have other defects that aren't necessarily related to the glycosylation, which is more for this protein movement. So giving us some insights into where the basic biology of what's going on in your child's cells is influencing all of their various manifestations. So then we talk about primary cells, and so these again are pieces of an organism, but these are naturally occurring, and they can often be obtained from using a small piece of skin to grow skin cells, or we call them fibroblasts. We can also use a blood sample to grow white blood cells, and when we immortalize these, they're called a lymphoblast cell line. And these are really incredibly useful CDG models. So again, the pros is that we can get these with a relatively simple office procedure, and they are a model of a CDG patient. In fact, they're a model of your CDG child when we use your child's cells, not just with your child's specific genetic mutations that cause that CDG, but all of the genetic material that they have in their cells that's exactly them. And we can use these to confirm new disease genes, determine what's going on in those cells, and then determine what's going wrong when those proteins aren't working. But again, it's not your child. It's just a model, and it can't tell you what's going on in the other cells of the body. It doesn't necessarily tell you what's going on in the rest of the body. And unfortunately, the process of obtaining those can leave a small scar. It is yet another medical procedure for patients who've been through a lot of other medical procedures. And it may not tell you about other genetic mutations in other children with your CDG type, but who have a different mutation. Primary cells have actually been a really important tool for Hudfries and his great progress in identifying these new CDG types. We heard about Fu8 and Bella's fibroblasts, and so that's what he's using to understand these new diseases. And he talked about this new understanding of Saul-Wilson syndrome, which is caused by a particular COG4 mutation, and understanding what's going wrong in those cells. So we've also learned about yeast as a model. So yeast is something many of you encountered today, Baker's yeast. We all have interaction with it, and this is really the simplest organism with a cellular process that's like the human, and it's a single cell organism, so sort of that next step from cell to organism. And this has historically been a good model to understand very basic cellular processes. So like cells, yeasts are really cheap and easy to grow. They're easy to do genetic manipulations, and we learned that yeast can be an important model because they can live with just one copy of the genetic material, so not like humans where we have a copy from mom and a copy from dad, but they can live with just one copy, which can make it easy to understand very specific single mutations. But they can also live with two copies of the genetic material, so we can use them to model how two different mutations are interacting together. And yeast are a good model for being used in high throughput screens. So of course, again, some of the downfalls of yeast is that as a single cell organism, it doesn't tell you about organs or the whole body, and that it's far enough removed from being like us that it doesn't necessarily have all of the same glycosylation machinery. So it does have limitations. And during the scientific conference, we've heard from Francois Folquier talking about manganese biology. This is particularly relevant to anyone with TMEN 165 CDG or SLC 39A8 CDG. And then we heard from Ethan Perlstein about using yeast to model PMM2 CDG patient mutations and using those for high throughput screens to identify compounds that improve growth and may improve outcomes for our PMM2 CDG patients. So the next model is worms. So this is often called C. elegans. It's a very particular type of worm that's really the simplest organism that has a nervous system. So we know that in the worm there's 302 neurons and there's 1,031 total cells in the adult worm. So not like the 3 trillion cells that Christina talked about in our body, but a simple way to look at an organism and many cells. So some of the, the benefits of the worm are that it has a developmental process that's really well understood. So unlike the human body, in trying to understand all 3 trillion cells, we understand what happens to every single one of those 1,031 cells. So it can really help us in understanding what happens in a multicellular organism because we understand it so well. They're cheap to breed. You can actually freeze them and thaw them and store them, and they're fine. So it's a convenient model for scientists. And then they have the ability that you can feed them a chemical that we call RNAi that can be targeted specifically for specific genes to use to generate some of these models. Now the worm doesn't have any organs, and it's still pretty distant from humans, so it has its downfalls. But we learned, again from Ethan Perlstein, about using worms as a model of PMM2 CDG, and as well as using a chemical inhibitor in that model to then identify compounds through a high throughput screen to overcome those effects. That has identified a new drug that has already resulted in an N of 1 clinical trial that we're all anxious to hear about how this drug, epalrestat, may be helpful for PM2 CDG patients. And then we talked about flies. So this is the fruit fly, so that annoying fruit fly when you leave your fruit out too long on the counter. In the science community, we call it Drosophila. So those are interchangeable terms. And surprisingly, the fly has a very similar gene structure to humans. The chromosomes are much simpler, so rather than the pair, the 23 chromosomes and its pairs that humans have, they have 4, so that can be helpful for genetic manipulations. They have a short generation time, which we heard about as being a big benefit of them, and they can be genetically manipulated. Now, of course, they're an insect, so they're not a mammal like us. There's still big differences, but it can be used as a good model. And we heard at the scientific conference from Clement Chow about using the fly for pig A CDG model. And he showed that because of the different cell types that are in flies, you can take this pig A gene out of neurons and cause a movement disorder, and you can take it out of glial cells and it causes seizures. And so this begins to help us understand some of that cellular biology of what's going on in these CDGs. We also talked about fish. So zebrafish, it's a freshwater fish. It's from the minnow family. You can get these as pets, and they are really useful models in that they have a rapid embryonic development. They have transparent embryos that develop outside their mother, so you're able to sort of watch that developmental process as it happens. They develop observable developmental behaviors, and you can use chemicals called morpholinos to turn off the genes. And then the fish also has a translucent body, so that allows you to visualize its organs and look at their function and their structure. Now you have to have a big water maintenance system for a lot of fish, and they're not mammals. And they still have a high genomic diversity and can be a bit more challenging than some of the other models to perform genetic manipulation. But during the scientific conference, we heard from Dr. Heather Flanagan-Steed about a PMM2 CDG model in fish that showed nervous system defects as well as jaw-shaped developmental defects of the cartilage. And HUD shared with us, again using a fish model for Saul-Wilson syndrome, and how it had some models of growth deficiency that may be similar to patients, as well as this abnormal bone development. And so then we have mouse, and so it's really not a wild mouse, but sort of a domesticated mouse that we've had with us for a long time in our human history. And it's one of the most useful model organisms for medical research. So there's a lot of pros. It's a mammal, so like humans, so it has a lot of close genetic similarities. There are a lot of tools for its genetic manipulation, and it has a relatively short lifespan, reproduction rate, and they're small, easy to handle, and relatively low cost compared to larger animal and mammal models. But of course, a mouse isn't a human. It doesn't really behave like a human. It has some alternative physiology. And what we've seen in all of the research that we've been doing with mouse models in other genetic diseases is that we're not always able to predict in humans based on what happens in the mouse. So there are differences. Christina talked about in her presentation some of the other disease mouse models and how they didn't necessarily have the same aspects of a disease as they were trying to model, which can make them challenging. And there are inbred strain differences between mice. So we did hear about mice during the scientific conference. Again, Dr. Taro Kinoshita talked about modeling GPI anchor deficiencies and showed a mouse that had seizures similar to what we may see in GPI anchor deficient patients. And Kent Lai talked about PGM1 CDG and a mouse model that he's working on developing. And now this meeting actually 2 years ago was the inspiration for some research that I'm currently working on. And that was at my first meeting here, heard a lot about the desire to develop a PMM2 CDG mouse model. And what we know about the mouse models that have been developed is that the first mouse model that was developed was a knockout trying to totally get rid of the PMM2 gene. And it turned out that that mouse, by basically putting the stop sign in the middle of the gene, was embryonic lethal. So that mouse died far before it could really develop very far. None of those mice could be born. And it really emphasized how important glycosylation is in early development and how critical of that process is for the total development of a complex animal or human. But in the discussion there, we know that we need good animal models to move our research forward. And so Misak and in Dr. Zin's session, there was a lot of discussion about the need to have a PMM2 mouse model. And so I went back to my institution and have partnered with the Amore Fund, as well as with the University of Pennsylvania and an investigator there, Dr. Jolyn Joe, who is an expert in neurologic disease, genetic neurologic diseases, and modeling them in mice, to work on generating a new PMM2 CDG mouse model. And so the goals we had in trying to develop this mouse model is to avoid that embryonic lethality. We need a mouse to be born so that we can use it as a model. And we wanted to make it relevant to as many PMM2 CDG patients as possible. So most PMM2 CDG patients have 2 different copies of their gene change. With many of them, and we've already heard this gene change mentioned, the R141H change. And then there's a second change. And so we decided to try and generate this model that has the second change that actually allows us to use other genetic tools to cause a cell type specific loss of PMM2. So rather than making PMM2 go away in every component of that mouse model, we can use the other tools we have genetically to take it out of just certain organs. So now, rather, we can move up from studying things just at a cell level and look at the organ system level and try and understand what's going on in the organ systems. And we're committed, once this model is fully developed, to making it available to other researchers so that they can study specific organ systems so that we can better understand really what is the role of PMM2 within the organs and how we can develop new therapies. And so this model that we've worked on developing will allow us to model a severe PMM2 deficiency, but in isolated cells and organs so that we can really do these pathophysiologic studies. And our focus, both based on Dr. Joel and Joe's expertise, as well as many of the primary medical concerns in PMM2 CDG being the neurologic aspects, have decided to focus on the neurologic system. Mouse models take a while. So 2 years ago, we started thinking about this progress and working on it. I don't have a lot of data yet on what our mouse model is doing, but the data that I am able to share with you is that we've so far had 6 litters of mice born. We've had 41 pups, and here, these numbers over here are the distribution of the numbers of each of those mouse babies that have been born with this model that we've set out to create. And the important numbers here are these 2 top blocks. So this block is those mice that are lacking PMM2 in their neurons, and these mice over here are those that are normal, and we don't see a difference in how many of them are born. So we've met our first goal quite reliably at this point of not having that embryonic lethality. And it turns out after I get back on the plane and go home, we're going to start all of our behavioral studies to identify if we have anything in these mice that is similar to what we see in our PMM2 CDG patients. So things are going well. It's a little bit slow, but I certainly hope at the next meeting or at the World Conference that we'll have some updates on what we see with our new mouse model. And so I'd like to end my talk by talking about what you as parents and advocates can really do for CDG research and for model development. So as we mentioned, fibroblasts can be a great model. So if you're interested, certainly the Amore Fund has been interested in trying to get more PMM2 CDG patients, and we're interested in getting this great cellular model that is reflective of your child's actual changes to be a good CDG model that we can use for additional research. And so interested families will be able to donate this in discussion with the investigators at the FCDGC sites. Beyond that and contributing to models, you can all consider participating in these FCDGC studies that Dr. Eva Mareva spoke about. And as was mentioned by Dr. Mark Patterson in all of our Doctors in sessions, we really want you all as family members and advocates to participate actively in our discussions of study design, outcome selection, so that we can make sure that what we're doing in our consortium is useful for you and for your children. And then certainly if you're interested in donating to research, CDG Care and the Amore Fund are both working really hard to raise funds to support additional CDG research, hosting fundraisers and all the things. So I definitely encourage you to stay active. You're all excellent in just being here, so thank you so much for that support. And I'd like to acknowledge all the support that we have from University of Pennsylvania, from CDG Care, from the Amore Fund, NIH, and for FCDGC and the Rare Disease Clinical Research Network. And happy to take any questions. This phone number is for my clinic site, so if you want to contact me through my clinic, you can certainly reach out. And this scan QR code will get you access to my CDG clinic site, and there's an online form for submitting questions as well if we don't happen to have a chance to meet up here. So thank you.